Okay. Thank you for joining us at REMAP Therapeutics today. This is our fifth, I think, fifth webinar. And today we have Ph.D. candidate Manesh Girn from McGill University. He's been lead author on multiple papers on the psychopharmacological action of psychedelics and the Cortex, as well as others. Thank you so much for coming today. The reason we're here is because you recently released a paper, which I'm scrolling to the long title of right now. Serotonergic psychedelic drugs, LSD and psilocybin reduce the hierarchical differentiation of unimodal and transmodal cortex. So
it's a mouthful.
Excuse my, I should have practiced that beforehand. So if you wouldn't mind, could you just tell us a bit about your background, what led you to this area of research in your studies and getting your, I'm assuming, a doctoral degree? So.
Yeah, for sure. So I've had a long-standing interest in psychedelics for various reasons, but it was majorly triggered, I guess, around 18 years old, 17, 18, something like that, when I came across Stanislav Grof's work. So he's like a pioneer in the space, specifically with LSD psychotherapy, good friends with Albert Hofmann and very connected to the whole movement, of course. And just fascinated by his work showing the kind of the range of states of consciousness psychedelics can induce, and including those that overlap with, mystical traditions and saints and yogis, etc. And I just found this stuff fascinating. And, I was shocked at how understudied it was and how multiple decades had gone with no interest, and how they were also at the same time stigmatized and demonized as if there was nothing positive that could be done with them. And so this motivated me to want to study it and try to understand, how psychedelics can induce these different states and how that can be used for behavior change and for healing and for therapy and this kind of stuff. And eventually, I've always been more interested in kind of the brain and the mechanisms that underlie the psychedelic experience and different aspects of our thinking. And so I got into cognitive neuroscience, and I used to volunteer at a lab during my undergrad. And then eventually now I'm in the last year of getting my Ph.D. in neuroscience, doing fMRI brain imaging research. So broadly speaking, my research is on the Deepamo Network. Actually, my dissertation is on that, independent of psychedelics, actually. Just trying to understand, what exactly is the Deepamo Network? How is it related to our cognition and perception and experience of the world? And I have a few projects ongoing on that. At the same time, I've been collaborating with Robin Carhart-Harris and others who have collected data already with fMRI brain imaging with people under psychedelics. So I've been analyzing, looking at these data sets to understand them in different ways, and also working with Robin and others to build more theoretical models of how they might work too. Once I've done my PhD, very likely I'll be joining Robin for a postdoctoral research position next year. That's kind of me.
Awesome. Well, I hope not to jinx it by saying congratulations now. So, but well earned. So just for people out there that perhaps are considering, that might be younger, that are considering going into this as a potential career path, which it is much more of now than it has ever been before. In high school, did you have a facility for sciences, or were you particularly interested in neuroscience back then, or anything like that?
Yeah, here's the interesting. So what I was interested in high school, well, first of all, at my high school itself, no, this wasn't really a thing at all.
Sure.
I was just a big reader. I would go off and read my own stuff. I used to read a lot about Zen and Eastern spirituality and meditation. And I got into psychedelics and the brain and the mind through an interest in philosophy, East and West, actually. And then just kind of pursued it from there. But what I would tell people is you'll be surprised at how accessible a lot of people in this space are. If you're like an interested, enthusiastic person and you've done a bit of your homework and you know what you're talking about, you can always, reach out to people through email, through Twitter, and even the top researchers, like they're just people too, and you can contact them and they'll respond a lot of the time.
It's shocking.
Yeah. Yeah.
I mean, that's been my experience for the last, 2+ years now. Just like Robin was like unusually receptive when I first came out of the study. Though I will say, I think having been a participant directly in a clinical university level study, it's a little bit like getting the golden ticket. And so people are kind of like, okay, data point, here he comes, so I don't know. And I think there's also just kind of there seems to be a team spirit of everybody is like, okay, if someone's been through a study, we're going to be there for them, shepherd them. Anyways, sorry. Yeah, that's completely been my experience. So
for sure, yeah. So like anybody interested in getting in the field, just first of all, I can't stress enough, do your homework, know your stuff, like sound like you know what you're talking about. Because like these researchers get tons of emails. Oh, I'm interested in psychedelics. Can I work with you? But if you don't come at that email with offering an idea or offering a reflection or setting yourself apart from everyone else, you're probably not going to get a response. So if you come across as an educated, enthusiastic person, just email the lab, not even like necessarily the top researcher, like the PI of the lab, the professor of the lab, email the research assistants, email the grad students, email all of them to get your foot in the door. And then once they start to get to know you, and it goes from there, and you have to build yourself up. That's basically what I did. Like, as I said, I haven't worked directly in a psychedelic research lab, but I've been very, very creative in getting myself involved. I contacted Robin with a research proposal in undergrad with my supervisor I used to work with over there, and built a relationship with him from an early point in time. And it's just like, flowered and developed over the years. But I created that out of nothing, and anybody can really do that if they're just, they go after it and take that initiative.
I completely agree. And I mean, I'd say for a lot of people, if they're talking about doing research, that means reading a lot of research studies and probably supporting literature around it. But it's okay, I think, to flail, I think, in there a bit, showing at least that you're willing to like immerse yourself in there. And it's like, after I think a certain prolonged period of that, it all starts to like make a more coherent sense. But yeah, that's great to hear. No, I have found multiple graduate students out there just like very eager and excited to talk. And it's like it's a shockingly small field, though it's growing. And then like in my particular arena around pain, it's even smaller, but even still.
Yeah.
So why do you have why is your focus on the default mode network? And can you explain what that is for people? I mean, we know, but for people that don't and are watching this for the first time, can you explain what that is and why it's crucial for psychedelic research?
Yeah, for sure. I mean, for me, the first question is why the Deepamo network in general, and I think it's a fascinating network because the network, the brain. Well, first of all, the Deepamo network is, to call it a network means it's a network of brain regions. So it's a set of brain regions that interact with each other more than they interact with the rest of the brain. It's like you could separate this cluster of regions that interact. And there's multiple, networks other than the Deepamo network, and there's various ways to define them, and it's a very technical area. But there seems to be this broad thing called the Deepamo network, which might be actually, composed of sub-networks, but let's not go there. Let's just say the Deepamo network. And what's interesting is the regions that compose it are the regions that are most expanded and more developed in humans relative to other primates. Our closest genetic cousin is a chimpanzee, and they have a lot of overlap with our brain in our visual cortex, in our sensory cortices. It's actually very similar. It hasn't changed that much, relatively speaking. But if you look at the regions which are now a part of the Deepamo network, these expanded considerably, like disproportionately relative to the rest of the brain. And these areas include the prefrontal cortex, which is a lot of people know that kind of layperson idea that it's involved in more complex thinking and planning and this kind of thing. So these kinds of regions, which are more typical of just humans, overlap with the Deepamo network. And also, as you might expect, then the Deepamo network is involved in a lot of the cognitive processes and types of thinking that are unique to humans, like our ability to kind of abstract from the present moment and think of stuff that's detached in space and time, imagine ourselves in different situations, and also our ability to understand what other people are thinking about and to think about their mental states. Also, our ability just for abstract reasoning and language in general is linked to the Deepamo network. And just like our ability to interpret and make sense of the world in this complex conceptual way is very much linked to the Deepamo network. And some would say, yeah, our sense of self is also linked to it, but a particular aspect of our sense of self, which is the one that's rooted in our stories that we tell ourselves about who we are, who we've been, who we're going to be, the kind of conceptual or narrative or autobiographical self, as it's called. So Diffusion Network is involved in all these very fascinating things that are core to the human experience. So I was like, yeah, I want to study this. It's pretty fascinating. And then obviously, when you think of psychedelics, they alter just the fundamental structures that we use to understand and interpret our experience. And so it would make sense that the Diffusion network plays some role in that too.
So there's these intimate, discrete types of self that are getting identified. So one of them that you mentioned was the biological self, right? So
autobiographical.
Sorry.
You mean?
Yeah, thank you. Autobiographical self, right? And what distinguishes that in contrast to other ideas of self? Can you be just a little bit more specific there?
For sure. I mean, I think you could think of perhaps three types, major aspects of self. Each one can be decomposed into more, but let's say three. So the autobiographical, as I said, it's like based in language. It's conceptual. It's our stories and narratives we tell ourselves and who we believe ourselves to be. But if you think about that, think about it. Even if you didn't have an idea of yourself in space and in time, or like conceptually, you still have a sense of being distinct from the environment. You have a sense that this is my body and this is a table. This is my body, this is the floor. This is my body, this is you. So like you could say there's like this other form of self that's been called the bodily self, or in some cases the minimal self, but that can mean something else too. But just our sense of having a body that's at a specific location in space. So they would say our autobiographical self is like a higher, more complex version that's scaffolded onto this more basic sense of I'm in a body. And this more basic sense, you could say, is shared with any animal, because in order to survive, you need to know what's you and what's not you at a very basic level. And whether it's conscious or not, that's a different question, but there's an implicit idea that this is me, this is the environment. And so those are two main parts of self. And you could say a third one, and this gets kind of like a bit more messy. It's just the basic experience that you're having an experience from somewhere. Usual human perception is we often feel like we're coming out of our head and seeing the world from there.
Right.
We're not just everything all the time, even though maybe we are, and some spiritual teachers will say you can get there. But, our usual human experience of the world is from a particular first person perspective located behind the eyes. So you can say that's another aspect of selfhood, probably the most basic aspect of selfhood.
So if we're talking about this autobiographical sense of self versus this bodily sense of self, is it safe to say that the autobiographical sense of self is a little bit more of the neocortex versus this bodily sense of self is a little bit more rooted in things like the insular cortex, where it's involved in this internal processing sense of internalized sensations and location, as opposed to say, and maybe you could also attribute that to stuff like interoception is what's primarily involved with insular cortex versus proprioception is a little bit more external, positions of your joints and limbs in space and relative motion to each other, but and maybe more directly involved in motion, which seems to involve more of that idea of maybe consciousness, maybe evolved as a survival mechanism to organize motion for dealing with threats in the external environment, right? Is that safe to?
To some extent, yeah, definitely. I think it also gets more complex. I don't think it's very hard to separate the neocortex from so-called more primitive areas. Like it's very almost fashionable to do that. But I think, for example, it's an open question to what extent our autobiographical self can be separated from our emotions and these more basic bodily experiences. Is there really something that's just completely conceptual and that's completely independent of emotion and feeling? And some scientists would say, no, it's all intertwined and you can't neatly separate them. The fact that we separate them is just a function of our language that we use to describe it, but it's not really like that. So that's one aspect of it. And in relation to the bodily sense of self, I think you're totally right. I think stuff like the insula, and the insula is very connected to your autonomic nervous system and the sensations coming from your viscera and all these other things. So I think that's definitely playing a central role. But in the neocortex, if you look at the parietal cortex, especially so-called parietal association areas, these areas integrate, our sense of vision with our sense of touch, and then with also insula, to create this kind of multidimensional, multimodal model of our body in space, basically.
Right.
And that's a cortical thing. So there's like different layers to it as well. And it's very hard to separate. I think the brain is such an integrated organ with the rest of your body, rest of your nervous system. I think if you try to isolate one thing and say it does this, it's going to break down at some point.
So just to be clear for people out there, so when you're talking about multimodal, multi-senses, it's not that they're encoding in your memories or encoding in your responses as discrete individual things. One is referencing the other and they're stacked. So often it is it's multiple stacked inputs, and that's how it's read, let's say by your conscious self. Once it's processed, like say through the thalamus and stuff like that, then you, that's been put through that relay station, then all these decisions are made, after everything projects back and forth between these different brain structures, then finally something emerges into the conscious self to act on. But they're dense packets from multiple sensory inputs, correct?
Yeah, totally. Yeah, something like that for sure.
Okay. So for psychedelic research, why does this default mode network play such a prominent role?
Right. I think first I want to say that, we don't know for sure that it does play a prominent role yet. And many will emphasize this, right? Because it is true, like for one, the studies to date have shown, sure, the Deepamo network is altered, but so are basically every other network in the brain. Some of these other networks more than the Deepamo network, right? The emphasis on the Deepamo network has been because a couple studies showed it was really evolution, for example, like the sense of our the experience of our sense of self-dissolving, but that wasn't replicated in actually all studies. And again, these other networks are just as altered. I think that the idea that Deepamo network is super important is because what I said before, because other past research has found that it's involved in all these very complex aspects of our thinking and perception. And so then we know that, those aspects of our thinking perception seem to be altered in a psychedelic state. Therefore, the Deepamo network is probably implicated. But as many would point out, this is something called reverse inference, which means you're looking at the brain and inferring a psychological function based on the brain alone, which is messy because a given brain region can do a multitude of things. So you can never neatly say, even with the amygdala, like, oh, amygdala was active. You can't just say like, oh, it's a fear response, because maybe it's activating in relation to something else. Because basically every brain region is like, I don't know what the technical word, I think it was pluripotent or something. It's like, there's many functions that can map onto one region.
Yes.
And so it's hard to, especially with the Deepamo network. So I would say it's very early stages in inferring this kind of stuff. I do believe that eventually the Deepamo network will be identified as a particularly important network for the effects of psychedelics. We just can't say that reliably right now.
Okay, so going into your research today, so I'm going to try saying it one more time. So this paper that you recently published, Serotonergic Psychedelic Drugs, LSD and Psilocybin reduced the hierarchical differentiation of unimodal and transmodal cortex. Can you tell us what that means and what you were looking at and what's come about as a result?
For sure. So let me start with the motivation for it. I think that helps contextualize. So, there's been a few studies looking at the brain effects of psychedelics now, of people in the experience and these are healthy people during their experience, what happens in your brain when you're just tripping and not doing anything in particular. And the thing with these studies is that, on a broad level, they have similarities, they show similarity, but if you go into the specifics, there seem to be differences. For example, on a broad level, you could say the brain becomes more integrated. A lot of networks, which usually are more distinct, are now talking to each other more. And the brain seems to become more complex in its activity, maybe more entropic, and these kinds of things. But then at the same time, it's like, if network A is more connected to network B in one data set, it might not be the same thing in data set two. It's like the specific network interactions that underlie it seem to be a bit inconsistent across studies, whereas the broad whole brain pattern seems to be similar a lot of the time. And this can be due to a whole variety of things, just because, obviously the effects of psychedelics on a person are so idiosyncratic. It's like very unique. And the experience itself is idiosyncratic over time. It's very variable. So it's like, obviously, if we were getting the same brain pattern for everybody, that would just be nuts. It's not realistic to even look for, especially when you're averaging across 8 to 10 minutes, which is what these studies do typically, not all of them. And so basically, the idea was psychedelics complex. If you look in the details, it seems to be finding inconsistencies. So how can we understand what is really real here? And so some people are saying like, yes, the brain becomes more integrated and it's more complex. That's what we know. So in this study, I wanted to see if we look at the brain as a whole and reduce the whole brain's activity to a limited set of patterns, how do these limited set of patterns change? Like, how can we collapse the complexity of the whole brain into, like, let's say 3 patterns? And this has been done outside of psychedelics with other research and just the normal brain of people doing nothing. And when they did this, when they collapsed the entire brain's activity into 3 patterns, let's say, the first, like, most significant pattern seems to separate these 2 types of cortex. So one is called the unimodal, one is called the transmodal, right? And so unimodal, what this means is like unimodal. Uni means like one, modal, modality. And what it's referring to is sensory modalities. So touch, vision, hearing, etc. So these are parts of cortex that are very much linked to our senses and process sensory information, right? So these are like very concrete, measuring, processing what's in the external world. So that was one end of this pattern. If you take this pattern to this other extreme, you get so-called transmodal regions. And again, trans means, like, you could say transcending, and then modal, modality. So it's going beyond just the sensory modalities. And the idea with these regions, which is supported by a lot of past research, is that these regions, which include the Deepamo network, are related to more abstract conceptual processes that are kind of a next step higher relative to just concrete sensory processing. So hierarchically, it's higher in the hierarchy. It's more broad and encompasses more sensory processes into it. And so, so past research has found that this axis from unimodal to transmodal is like a core organizing principle of the cortex. The cortex seems to be separated from one to the other. And when you process sensory information, it seems to go up the hierarchy and then like back down and so on. But like this seems to be a pathway in the brain, a processing pathway through which stuff comes from the senses and eventually turns into beliefs, concepts, stories, this kind of stuff. And so usually in the brain, these 2 areas are distinct from each other, as you'd expect, because you want to have your senses and your experience of the external world separate from your concepts, beliefs, and internal thinking. And that makes sense, right? And so what I wanted to do with this study was apply this analysis that found that distinction between unimodal and transmodal cortex in normal people without drugs on a psychedelic to see, do these 2 extremes come closer to each other? Is there less distinction between this abstract high-level regions and these concrete low-level regions? And so that's basically the gist of what I did.
And those have been, so unimodal versus transmodal. So unimodal, if we're talking sensory data, that's also referred to as bottom-up processing, as it's coming from the bottom of our senses, out in the limbs, out in the peripheral nervous system, down in the trunk. And then that's ascending, that's afferent nerve signals, so going upwards towards the brain, right? And then top down, or the transmodal or transcendent, we're talking abstract, conceptualized thinking or perceptions. And that's in fact top down or descending thinking. So that's stuff that's, once it's received the sensory information, it's integrated it, it's processed it, it's made its conclusions, literally, right? And then that creates a perceptual, either a perceptual output or a motor efferent or a descending signal that's like, okay, make this muscle fire or accelerate the heart rate or decrease the breathe, whatever it is, right? So in part, so there's a perceptual component, and then there's like generally a motor component. Is that safe to say?
Yeah, no, that's a good way to think about it. Yeah, definitely. It's like, yeah, it's like it goes up this pathway, and then once it gets up there, that can later influence our perception and sensory processing by biasing our attention. Or as you're saying, yeah, it can lead to certain actions that are different and interpretations of experience and so on. Yeah.
Okay. So what did you guys do?
Right. Yeah, so let me describe, I can describe the methodology in more detail, but one thing I want to bring into this is important, is the Rebus model.
Right.
Because another motivation for this was to test the hypothesis of the Rebus model. Now, the Rebus model is the relaxed beliefs under psychedelics model proposed by Carhart-Harris and Carl Friston.
Right.
And, the core idea there is that, to put it in a nutshell, is that they're proposing the idea that psychedelics elicit their characteristic effects and therapeutic effects because they kind of destabilize these priors or beliefs or assumptions about the world and who we are, such that we're able to now take in more information to update them and revise them. So we usually, view the world through this filter of beliefs and concepts, and usually we don't know that filter is there. It's just there, and psychedelics kind of show us that it's there, make it less stable, and allow us to change it in potentially healthier ways if we in the right context and so on. And so an hypothesis from that is that these high-level areas, transmodal areas that we just discussed, are the ones that encode these beliefs and assumptions, right? And usually they're pretty distant from our incoming inputs and can be kind of crystallized into patterns which are hard to shake.
Right.
And so one of the hypotheses was that, in order to make these beliefs and assumptions less stable and more sensitive to incoming inputs, they have to be less differentiated from the inputs. And so that's what the study comes in. It's like, I'm seeing, are these low-level inputs less differentiated from these high-level ones, such that they're exchanging more information, and perhaps the high-level ones are more susceptible to being influenced by them.
Okay.
And so this was also a test of Rebus in that way.
So we also, besides referring to like this unimodal activity or this bottom-up sensory processing, it's also known as error propagation, right? Or error correction propagation. Where you have, so in the transmodal, you have this top-down processing where we have these crystallized beliefs or frameworks or values, right? Or self concepts, right? And you referred to them as a prior, they're known in the Rebus model as a strong prior. So it's like it's there before, and it's been there repeatedly to such a degree that it's persistent, right? But the idea is the unimodal or the bottom-up sensory processing, it's supposed to be passing in new sensory data that either validates that strong prior or says, hey, no, that doesn't really match what's going on out here. You think this, but ta-da, this new information says that's actually not 100% correct. It's only 72% correct. Let's adjust that, right?
Yeah.
And then if we're talking like a strong prior in a person with, say, severe depression or something, it's like, I suck, or nothing ever works out for me, or I'm worthless. Things, that type of lovely iterative rumination that has such a strong undertow to it that you don't even recognize that it's operating, and yet it shapes all decisions and perceptions going forward, correct?
Totally, yeah. That's the idea for sure.
Yeah, lots of fun. Personally, yeah, I'll just say, and that's also something we could refer to if we're looking at chronic pain, right?
Mm-hmm.
As so, this is known as central sensitization, as in sensitivity that is generated out of the central nervous system, and it's
Mm-hmm.
also a strong prior where even though the tissue damage has completely completed its healing.
Mm-hmm.
There's no more noxious or threatening input coming from the limb anymore. But at some point, there was this pain neurotag that was formed, and now it's embedded. It's a strong prior, and there's nothing countering it. So you're, if we're talking fibromyalgia or phantom limb pain, that's also persisting, correct?
Mm-hmm. Yeah, yeah, that's one way to think about it. Yeah.
One way to think about it. Sorry, correct is maybe not the best word. So please keep going.
Yeah, yeah. So I mean, so so to foreshadow the results or to say the results, we did find support for the Rebus model, and we did find that these areas of these kind of distinct areas of cortex were becoming less differentiated from each other, which does suggest that in the psychedelic state, the brain is less able to differentiate our sensory processing from these high-level priors and beliefs, and so on. And now, we can't necessarily infer directionality. We can't infer that now sensory areas are affecting transmodal more or transmodal are affecting sensory more. It could be either one. It could be both. That requires more research. All that we can say from the study that I did was that they seem to be becoming more interconnected with each other. And what's interesting, moreover, is that this specifically was related to these kind of sensory areas becoming less integrated with themselves and kind of more integrated with transmodal, whereas transmodal stayed connected with each other, but just became more connected to the rest of the brain. So it's kind of different on each side in that way, but basically it seems like there's sensory disruptions, and they're giving more precedent to this global transmodal pattern in some sense. But it gets messy. There's a lot of there's like alternative interpretations here. Mhm.
Which is something that we talked about previously on our webinar. So you did, so these come, so there's, okay, I've heard a couple critiques, so which I'm just learning of and barely understanding, and you seem well situated maybe to discuss some of it. So this study is an analysis of two previous data sets that were done, I don't want to say years ago, but let's say a little more than half a decade ago. One was involving LSD, one is involving psilocybin, right? And people that took it one time or went into the scanner once under a placebo, so they didn't know if they were going to be getting LSD or psilocybin, and then the next time they go on under, they received one of those agents, right? And roughly it's a group of I think 9, and the other one was 15 after it kind of shook out and everything. Like, and there were some people that were excluded because of one reason or another. These were resting state analyses.
Yes. Yeah. So sorry, you can go ahead.
Go. No.
No, I was gonna say, yeah, no, this is a good point. I need to go back and to say exactly what we did. Yeah, so we had 2 data sets, one with psilocybin, one with LSD. Pretty small data sets, but that they were just all that's available because this stuff is expensive and hard to do still. And they're having previously published, and I was just looking at applying this new metric or analysis of the brain to characterize it in a different way, because I thought it would be informative, for the reasons we've just been talking about. And when people took it, it is technically a resting state, in that they given the drug and then were sitting there not asked to do anything. So it's very unconstrained, and so it's really subjects in both placebo and the drug conditions are able to just do whatever they want. Like, we're not controlling what they're doing.
Right.
And so in this sense, it's like how I see it is a resting state, quote unquote, resting state study like this is like trying to see if we take a snapshot of this particular state of consciousness or way of being, and a snapshot of what their brain is doing, and that we hope accurately represents what it's always doing in that state, and then compare that with a different state. What are the differences? So we're basically asking, when somebody's just sitting there doing nothing, what's different when they're sitting there doing nothing but on a psychedelic? And sitting there doing nothing could be a whole variety of things. It's probably mind wandering, daydreaming, going back and forth between thinking about how annoying this scanner is and thinking about what they're going to do later, or whatever. And it's very uncontrolled. But again, the purpose of it, I think, is to try to get a representative snapshot of that state of consciousness in general.
So when you say representative snapshot, it's like some people like have argued like, okay, resting state, it wanders so much that who can get an accurate picture of what's happening? But you are saying the picture is of a wandering, non-constrained mind. When you are just absent-mindedly not doing something, that is in itself a snapshot that we can like look at and identify. It's like, oh, look, this is like a wandering mind. He could be doing different things, thinking about different things, but it's not directed towards a specific task at that moment.
Yeah, totally. And past research has found that in high quality data, you can take somebody's resting state brain, scan them for 15 to 20 minutes, and then scan them again, a different day, and it correlates at like 0.90. It's like, it's a very strong correlation on different days of that same person. And so even though on each day they were thinking about different things, they're not can do the same things at all, there's some consistency suggesting that it is catching something that's like intrinsic to the person. That's the whole idea. And there's like hundreds and, probably thousands of studies looking at resting state in a variety of ways.
And intrinsic to that pattern of cognition, that pattern of thinking, right? Like that resting state has, when you look at it, it's like on a scanner, it's like, that looks like resting state. I mean, more or less.
But what's interesting is that if you give somebody a task or get them to watch a movie, it's very, very similar. It's not completely different. It's like still a very high correlation. So there is consistency across states, like very high consistency across states as well.
So can you talk about this new method? It's called, and I'm sorry, I've highlighted so much, gradient, what's it called? Gradient?
Yeah, gradient mapping, you can call it generally.
Gradient mapping, yeah. Great.
It's the actual analysis is called diffusion map embedding, which sounds a bit more crazy, but it is some technical stuff. So, I mean, I can try to describe it. So we start on one end, the brain, right? And what these fMRIs do is they separate the brain into a whole bunch of, three-dimensional like cubes. You could say the brain is just like a whole bunch of cubes in brain shape, square cubes, but in the shape of the brain. And then you reduce that into brain regions. So a brain region is just a bunch of cubes that are next to each other and which have activity patterns that usually correlate. So you take the brain, let's say you turn it into 400 regions, and then we look at how does the activity of these 400 regions correlate with each other over time, right? It's like how similar are the patterns, the spiking patterns across regions. So then you get this what we call a matrix of the connections, the correlation between every region and every other region. And then what we want to see, what I do with this method is how similar is one region's connectivity with every other brain region to another brain region's connectivity with every other brain region.
Okay.
So then what that gives you is a matrix of similarity between regions. So then, you could think of a big giant square, 400 rows, 400 columns. Every element within that is how similar is this region's connectivity with the rest of the brain to the other regions, So it's like, we call it functional connectivity similarity.
Okay.
And then based on that similarity, we're feeding it into this diffusion embedding algorithm, which is what we would call a dimensionality reduction technique.
Okay.
And what that means is that this square that we just talked about is 400 by 400. That's like 160,000, right? It's a lot of things going on. And what we want to do is collapse that into the most dominant patterns that are spread across the whole matrix.
Okay.
And so what this thing does is basically it's like, what are the main dimensions of similarity that capture all these regions? And so then we use that, and that's how we get this gradient, or the dimension from unimodal to transmodal cortex. It's saying when you look at the similarity of all the regions in the brain, the most dominant pattern is one that groups regions that are similar on one end and similar on another end, which are different from each other. So what are like the anchors of similarity that are dominant in the data? And then what seems to be the case is one anchor is transmodal areas, one anchor is unimodal areas. And then there's like a whole spectrum of similarity that goes from one to the other. I hope this makes sense. And so it's like a dimension of functional connectivity similarity from these unimodal sensory areas to transmodal abstract. And then what we're doing is basically asking in a placebo state, how present is this dimension? How like accentuated or salient is this dimension in the whole brain connectivity pattern? And then how much is it also present in the psychedelic one? And then we see it's much less present in the psychedelic one, meaning that there are less, these opposite poles of this, or anchors, are less distinct. Like this whole dimension is being collapsed a bit. This is where you get the concept that, oh, we're collapsing or flattening the hierarchy of the brain. Right. There's this hierarchy from low-level sensory to high-level abstract. And so that's what we did. And we found that the gradient is indeed contracting, which is why we're saying unimodal and sensory and transmodal are differentiated. And we took that, actually, I took that a step further because I wanted to see, like, what does it mean that they're less differentiated? You know, what is leading them to be less differentiated? Then I ran some additional analyses, which I won't get into the details of these, but then they showed me that what happened was the sensory areas, which are usually very integrated, became less integrated with themselves and more integrated with the other side, the transmodal. And so like we're like kind of almost like altering the fidelity or kind of modularity or segregation of unimodal areas, and now they're blurring more together with transmodal. And this is on the higher level, seeing as a collapse of this hierarchy. And so that's, yeah, I hope that makes sense.
So then when we're talking, so when we're going from an analysis of the mechanisms of action there going on within the cortex and within that cortical hierarchy, right? And we're you know, okay, here we have the transmodal versus the unimodal. So top down versus bottom up, just to really abbreviate. But when we translate that into like the actual psychedelic experience, so people talk about this, oceanic boundlessness, the sense of oneness, the sense of unity, but also this sense that like, my sense of perception proceeds outward to infinity. Like I can feel the stars vibrating, my ancestors are, I can hear them breathe, stuff like that, where there's all this stuff that's normally differentiated, separate, cleanly and clearly separated. Now it's like, sense of time is gone, sense of timing, sense of self is like, you're talking about things collapsing, things that normally give us, I don't know, a sense of triangulation on reality. Are kind of, okay, now we're looking at fewer data points. They're all kind of starting to merge into, is that some of what we're seeing here?
Yeah, you can say that. Yeah. Ultimately, we need, data sets which actually collect data on some of this stuff. It's like asking people these questions in some way and then correlating that with the brain to see how it relates specifically. Since we can't really do that with these data sets, we can only speculate that something like that is going on, right? And it is very possible. But I also think, in order to have ego dissolution, you don't necessarily need to have the whole brain integrated. It could just be those central regions, which are usually most important for maintaining our sense of, individual self, which is a carefully constructed, illusion in a sense by our brain, right? And so like, it doesn't take the whole brain to create that. It just creates the regions involved in spatial location, the regions involved in our memory of ourselves. And if those are gone or disrupted, then we can have this experience without the whole brain becoming de-differentiated. But I do think this result is interesting because it does show that this fundamental dimension around which the brain is organized seems to be altered, and it's pretty stable otherwise. Like this is the first time it's been shown to be altered like this by a drug. And even for example, people suffering from autism or like very old people, if you give them this gradient analysis, if you run it on them, you find much less differences. But what is interesting is you get similar stuff with schizophrenia.
Right.
Patients with
I saw
schizophrenia.
that towards the end of the paper.
Yeah,
And obviously, or maybe not obviously for people that are listening to this, like LSD, when it first came out, was characterized as a drug known as a class of drug known as a psychotomimeticist, right? Mimicking psychosis. And it was initially thought that therapists could take it to get insight into what their schizophrenic patients were going through and maybe how to better offer them solutions to what they were dealing with. But not quite that viewpoint anymore on psychedelics, is it?
Yeah, for sure. Yeah, it didn't last very long. Is even in the late 50s, early 60s, they realized that people were having wonderful experiences. They weren't psychotic, and it's just like psychedelics are. But I wouldn't say it's like they are not able to do that, and they're this. It's like it's always it's everything. Depending on context and what we know now and setting and all this.
So if we're seeing this de-differentiation between the transmodal and unimodal cortices, if I've got it right, what is the speculation now? I mean, though obviously the Rebus model and others posit this, and then I just remembered something about other models that I just wanted to ask your opinion on. Why is this seen as having this therapeutic benefit? Why is that de-differentiation? Why doesn't it I asked this to Robin one time. I'm like, okay, so we're increasing the entropy or the chaos or the degeneration of organization there in these hierarchical structures, right? Why is it that you go through this psychedelic experience and then when you're coming out, it suddenly resets into this new effective and for many people healed, I hate to use the H word, but or you can even go as far as with the C word for some people. I mean, it's astonishing how fast that remission will set in. Why?
Right.
Why?
Yeah. I think a few points here. One, I mean, it doesn't happen for everybody. It's it doesn't always happen. And it really depends on, no, for sure. And it really depends on how the experience is supported and where it goes, Obviously, you can have a terrible experience at a music festival and you're not going to come out of that being healed or cured or whatever. You might even just be like traumatized by that to some extent and, be psychologically destabilized or something.
Yeah.
So I think what they do is they kind of, and this is like a cliche, I hate to say this, like shake the snow globe. I don't like saying that. Let's just say it perturbs our regular mode of functioning. It releases us from our habitual frame or myopic view of the world and ourselves and just pulls us out of that for a little bit. And so then we're able to see things from a bit of a new perspective because we're just not in our normal frame of mind. And in that shakeup, in that kind of shaking from our psychological mores and our tendencies, we're able to then come back into a new frame of reference, slightly altered. By shaking it all up and making it kind of less rigid, that opens the opportunity to change what was there. Because it's very hard to be, even if you think about in your life, if you go through this huge shakeup in your life and everything is shifted, when you come back to your usual everyday, you're going to be slightly different. You're not just going to come back with the same thoughts and same mental headspace. So you go on a trip around the world and come back. It's, it shakes up your frame. And I think psychedelics do that. And so by the kind of like inducing this entropy or chaos temporarily, you create an opportunity where you could then step into a new mode of being afterwards. And if you're properly supported. But the important thing is that this mode of being is not going to last forever magically. So you got to come into that and then notice how your brain is going to try to pull you back to that over and over and over again. And you have to consciously say like, oh, this is a new way and I like this, it's healthier, it's better. And you have to integrate it and, continue that.
Okay, so there's a process of reevaluating all of these frameworks that we have, right? You go on this, like, say, like a global journey or whatever, or you have this major shift in your life, some type of lifestyle change, and then you're reevaluating all the previously held beliefs or perceptions in a way. And then it's like, what is my return of investment on this or something like that? Or gosh, I've had this very biased view of that and it's been really skewed and off. I haven't been thinking about that in the straight way at all. So is there an inherent, almost endogenous default setting you can restore to that is like, say, like clean or non-biased, and when someone is being shook up like that, it's very sensitive to outside conditions. It's very sensitive, to the setting you're in, obviously, and to your mindset. So it's this classic set and setting, but sorry, I got about 3 steps ahead of my train of thought here. So I'm working my way back, but why? Okay, let's just go to the first simple one and it'll come back to me. Is there a conceptualized idea that there is this like unbiased default that's operating within the neural structures?
Right. It's a really interesting question. I mean, it depends who you ask, I think. And it's hard to say it from a brain's perspective. That, is there like some pure version of yourself that is able to experience things clearly? Talk to any mystic or a spiritual person, they'll say, yes, you can get there, meditate enough, kind of develop beyond your cognitive and emotional biases, attain the centeredness within yourself, and you're able to see things clearly.
Remove
that's
the scales
Zen.
from your
That's
eyes.
It's whatever. Yeah, exactly. Yeah, that's like, standard Zen kind of philosophy, and also other traditions too.
Sure.
Which very well could be possible. Like, I'm not saying that it's not. So I think it is possible to allow yourself to, attain this greater disidentification from the mind and all of its structures. The structures will be there, but it's like, how much are you letting them dictate your perception, emotion, thinking, attention, most importantly. So I think, it is possible to be in a place where these things are ongoing. Your mind is saying its thing. It wants to bias you, but you're able to separate yourself, your attention from that, and live in this more pure way. And I think that's what spirituality is to me, or mysticism.
So you also mentioned that you can't go into this state and like just let it maintain on its own, that you have to do this so the second part, this so-called integration work, right? Mm-hmm. That it can't happen on its own, but it seems post-dosing during this neuroplastic window, interventions that previously had no traction suddenly become far more impactful and much easier to implement and almost rewarding in and of themselves. I mean, we could look at anhedonia in the role of all of that. So hedonia, inability to feel pleasure, right? But why? I mean, I have lots of thoughts about this. I have a lot of thoughts about this, but why would psychedelics suddenly make all post-dosing interventions far stickier and more salient or more meaningful, positively meaningful? Why? Sorry.
Yeah. So the short answer is we don't know, right? Of course. But I think interesting. It's a very interesting concept, and it's multiple potential ideas on why. One is kind of what we were saying before. It shakes you up in a kind of puts you in this more open and receptive state to the world, and to kind of exploring novel ways of being and thinking and et cetera, behaving. I think that, just kind of afterglow effect of the psychedelic will make you more open and sensitive to these other interventions. The other side of this is just like boosted neuroplasticity, like your brain has more potential ability to create new connections or to rewire old ones, and that lasts is sustained after the acute effects, and therefore you're just a bit more able to create these changes in your thinking that were kind of more difficult before, especially because, you know, if you're depressed, you're suffering from chronic pain, chronic stress usually, and chronic stress is terrible for neuroplasticity and just atrophies parts of your brain and so on. So
Oh yeah. No, I mean, so I mean, I've said this before, but like my first glimmer of hope in the study was I read the Lee paper from 2018 on dendritic arborization and neurogenesis, synaptogenesis, and like, you know, all of those things are things I had been studying from a chronic pain perspective for, you know, a decade and a half before through my certification organization, you know. So we're studying neuroplasticity for like changing chronic pain, but I had completely lost track about the dendritic atrophy that occurs in the prefrontal cortex that chiefly characterizes depression. And it's like, oh my God, you know, there's this hole literally in my head. And and so it was my first real sincere burst of hope outside of like hoping that the experience itself would create a psychological reframing. But I wonder if we're also talking about this from a psychological perspective, or from this reframing perspective, and this you know, decreased differentiation. It's a little bit to me like when people are looking for a new job or something, they say don't talk to the people that are the closest to you. You have to talk to the people on the periphery of your whatever your social network is, because they're going to have information you don't routinely interact with. And that's where some novel opportunities or viewpoints might, you know, oh my God, I had no idea that this job was out there, you know, something. Is that perhaps a similar idea to what's happening in here?
Right, with psychedelics in general, you mean? I think that's a cool analogy. I think, yeah, I mean, we're going beyond our circumscribed space of mental states, you could say, and going out of that to fetch new insights and things. I think that makes sense in terms of what psychedelics can do. Thinking, yeah, I think psychologically that's one of the main things that they do. Yeah, I would agree with that.
We've been going for a while here, so we should probably wrap up here. I just two quick questions. Just one, what were the main differences between the LSD set and then the psilocybin set? And I know you had some problems just because the psilocybin set was smaller, so there's just they call that, I guess, underpowered, right?
Yeah.
So can you talk a little bit about the differences you saw between those two? I mean, right away, I just speculated. I'll just say it's like LSD is neurochemically promiscuous. That means it's operating on more neuroreceptors, neurochemical receptors. So just more activity in general, but.
Yeah, totally. I mean, yeah, it's unfortunate that these samples are so small. So it's hard to say whether a difference is due to actual drug effect or noise or, you know, or motion, or just like the, unreliability of any measurements based on a small sample like that, you know. What we did find was like, for one, they're remarkably similar, psilocybin and LSD, in the overall pattern. And I can say from other analyses which I've done with DMT, it's pretty much the same thing with DMT, except DMT is just like these drugs on steroids. It's just much more intense. Like the effects are much larger. So I think across these three drugs, three distinct data sets I've looked at, it's a very similar pattern, which helps me gives me confidence in them, even though they're small data sets. If you see it multiple times, probably something's there. To come back to this study, we found that LSD had more effects on visual regions. And you see this. We didn't talk about the other gradients. So there's two other gradients as well that I mentioned in the paper, and in one of them, it's you know, visual network, visual regions are very important, and you see much more differences in LSD relative to psilocybin. And this could be due to a whole variety of things, but it's consistent with past results too, showing how LSD really changed the connectivity of visual areas, whereas that wasn't found as strongly with psilocybin. And whether, you know, you can say from this that LSD is just more of a visual drug, like you can't really say that. depends on dosage, depends on so many
Well, I was
things.
going to
And
say
so,
that's hard to believe because
yeah,
25 milligrams of psilocybin is very visual, at least if you've got your eyes closed. So,
definitely. Yeah. So I think we can't really make claim anything about differences between these drugs based on these data sets.
okay. Um, so then the other thing would, I guess, just going to, let's say, just critiques out there about maybe the Rebus model itself, where like some people, have asserted, and this is all new to me, that when you're talking about top down versus bottom up, that it's not so clear cut that like when you guys reference that the you and everyone else in your paper, the other authors, you reference, well, hey, this isn't all you know uh cut and dry, that there are other models out there like thalamic gating, and that so that there's so many projections going back and forth between the sensory cortices and these more abstract top-down cortices, and that it's not a one-way flow, that there's a ton of interaction and like so-called projections from one area into kind of like your 16,000 square cube, like it's like everything is relating to literally everything else. And so the main critique is like, okay, Rebus has got a great narrative. It's rapidly comprehensible. I mean, it was, once I had it explained to me, it's like, oh my God, this gels with my experience. But then you hear about these others, it's like, oh, holy cow, that's more complex, but that does kind of match maybe other parts of the data. Can you talk about that a bit?
Yeah, it's definitely,
Sorry.
I mean, even if you talk to Robin, he'll say like, this is just like, it was a theory based on the data to date. It's
Right.
a lot of claims that are not empirically verified necessarily. The strength of it is that it synthesizes a lot of research. It has a good intuitive narrative. I think it did get probably some things right, but there's definitely a lot more to it. And certain things might be flat out wrong that he predicted in there. So I think that's important to recognize. And yeah, I think in relation to other theories, like for example, I know Manoj had and Fred Barrett and them had their claustrum-based theory, and I think the claustrum is very important, which is this little thin region in the brain. And then also going back a long time is Vollenweider's theory of thalamic gating and applying that to psychedelics as well. And I mean, I think I mentioned this in the paper towards the end. It's like they're not necessarily mutually exclusive. It could be all of them.
Yeah.
Like for example, this paper and is actually only including the cortex. It doesn't. I didn't include subcortical regions because this measure hasn't been applied to subcortical and it doesn't really fit into the framework. But obviously, subcortical ones are central, and it's very likely the case, if not, definitely, well, it definitely is to some extent the case that these changes in cortex are the result of subcortical areas affecting the brain, the cortex, including the thalamus. So thalamic gating is basically the idea that these thalamic connections to the cortex are becoming disinhibited, and now they're overflowing the cortex with sensory information, and this overloads it, and it leads to this breakdown and disintegration. And that could be consistent. It could be these thalamic connections could be leading the brain to organize in a different way, such that these hierarchical areas are less differentiated. And same with the claustrum. The idea is the claustrum regulates brain networks and their interactions. So you alter 2A receptors, serotonin 2A receptors that psychedelics hit on the claustrum, and it's going to affect the cortex. And so what we're seeing on the cortex could be the consequence of these lower order claustrum and thalamic changes. But again, we need more data for that. And one thing else I want to say here is that the thalamus and claustrum are very hard to analyze reliably in the brain using fMRI. They're often very noisy, more susceptible to motion and all the issues. So we need larger data sets with high quality data to really reliably say anything about those regions. And I didn't really think it could be afforded with the ones that I used.
Fair enough. No, you have to make a choice about what you can like reliably discuss. Is anybody looking at the contributions of the PMRF or the cerebellum, so the midbrain or the hindbrain, and like what's going on there? I mean, I could go on all day about, what their interactions with descending inhibition, and like central structures, as well as accuracy, balance, and coordination, not only of motion, but perhaps of thought itself and perception. So I would be, is
Yeah.
anybody kind of digging in there?
Not that I know of, and even in general neuroscience and brain imaging, these are neglected areas a lot of the time. And because one of the reasons is because when you, in the normal ways that we use a scanner to scan people's brains, we focus on cortex and all these other areas, and subcortical areas and the brainstem take a hit in how accurate readings of them are. Because in order to get accurate readings of them, we have to sacrifice and not measure aspects of the cortex most of the time. So this is why it's like you have to be very, very interested in just those regions mainly in order to do it. But there are technological advances that are making it better. But historically, that's why they're kind of understudied because of that.
Okay. Is there, we gotta, I don't want to take up any more of your time. Anything you want to say in closing? Anything you'd like to emphasize or re-emphasize?
Yeah. Something I think, and this is implied by this study and my reasoning behind it, is that psychedelics can be more so characterized as affecting the entire brain and changing just the fundamental ways in which the brain functions. And I think rather than looking at specific networks and just honing in on, oh, is this network connected to this network? And is that the case in all the drugs? It's like going up a level and saying, what are the general characteristics of the whole brain that are changed? It's more complex. Maybe there's unimodal, transmodal de-differentiation. Maybe there are other metrics which capture the general mode that the brain goes into. So I'm moving more towards understanding the brain, and eventually the brain in constant interaction with the nervous system, of course, and body, as this integrated unit that we can really understand in a very informative way if we look at it at like a meta whole brain, whole nervous system level, rather than zeroing in on specific networks. So that's kind of abstract, but I have a paper me, Robin, and others are writing along those lines. So yeah.
Okay. Well, we can't wait to see it. So just thank you again for being willing to do this today. Appreciate it so much. Can't wait to see how this further develops. And, I'll be continuing to kind of go back to this just to see how it would apply to, things like chronic pain and potentially this. I'm very interested to see what's going on with the unimodal aspects. So obviously for that same reason. So thank you again so much for joining us today. And before I go, everybody, remember please go to remaptherapeutics.com. We have a newsletter where we talk about our webinars that are upcoming, but in particular, we have our first public psychedelics and pain symposium with noted panelists in psychedelics and pain research from around the world. That's coming up June 25th at noon till 4:30 p.m. We also have some other announcements coming out soon, so keep an eye out for that. And we try to maintain a schedule of about one webinar a week with guests, much like Manesh here, who are doing leading edge research in this field. So thank you all, and talk to you soon. I'll end the recording here.